In this experiment, we will be observing how E. coli evolves, how E. coli resistance to ampicillin evolves, how soil bacteria reacts to ampicillin, how soil bacteria evolves in general. Doing so involves growing E. coli and soil bacteria in different test tubes, 2 test tubes containing E. coli bacteria, one growing in LB only, while the other consists of LB and AMP. The same is for the soil bacteria, one in LB only, the other in LB and AMP. The conditions involved with the evolution of antibiotic resistance is mutation, which is mutated strain of bacteria is more resistant than the original strain, and selection, where the resistant strain survives the antibiotic sweep and reproduces to have more resistant offspring.
Here is the background on the organisms. E. coli is a gram-negative bacillus and is a part of the gut biome. Virulence determines the disease strength and its ability to build up resistance to common antibiotics. Different strains have different effects, like watery or bloody diarrhea, urinary tract infections, pneumonia, gastroenteritis, renal failure, and sepsis shock. E. coli's main form of attack is by creating toxins when attacking the cell wall. Background on ampicillin is that it is a penicillin class of medication used to treat bacterial infections. It's also beta-lactase antibiotic and is in the aminopenicillins. The mode of action for ampicillin is through PBPs, penicillin binding protein inactivation. PBPs maintain the cell wall, and without it, the cell wall goes through lysis and cell death. The main advantage of using ampicillin is that it's resistant to acids, meaning that it won't be broken down in your gut. Ampicillin resistance in E. coli is from beta-lactosomes that are produced by E. coli. These bind to primary receptors, which then affects their function, causing cell death. This inhibition causes a resistance to the antibiotic because it inactivates ampicillin, allowing for the infection to persist.
These are our hypotheses and predictions. For LB cultures, our hypothesis was that E. coli grown on LB will show normal growth, while E. coli initially grown on LB and then plated on LB with ampicillin will show little or no growth. The predictions for this are that the LB plate will have many colonies that should be observed, indicating normal bacterial growth. And our LB plate with ampicillin will have few or no colonies that should be observed because the ampicillin will kill most of the bacteria. For LB with ampicillin cultures, the hypothesis was that E. coli grown on LB with ampicillin will develop resistance and thus show growth on both the LB and the LB with ampicillin plates. Our predictions are that the LB plate will have many colonies and the resistant bacteria will grow without inhibition. The LB with ampicillin plates have many colonies that should be observed, as the bacteria are also resistant to ampicillin. For culture plates on LB plates, our hypothesis was that E. coli grown on LB and then placed on either LB or LB with ampicillin will show growth on the LB, but little to no growth on the LB with ampicillin. Predictions were that the LB culture plates would have many colonies, but the LBs with ampicillin would have few or no colonies. For soil bacteria with LB culture plates, our hypothesis was that the soil bacteria grown on LB will show normal growth, while bacteria initially grown on LB and then plated on LB with ampicillin will show less growth. However, the soil bacteria will grow more than the E. coli plates on ampicillin. Our predictions were that the LB plates would have many colonies that were observed, indicating normal bacteria growth, but the LB plates with ampicillin would show less colonies. However, there shall be more bacteria present than in our E. coli samples.